Hello and welcome to part 2 of our lecture on blood. In this portion we're going to talk about clotting and blood typing. And so we're going to begin here with this idea of clotting. What exactly is a clot? How does this happen? Well, clotting is going to obviously protect a damaged vessel. So when a vessel is damaged, we're going to have platelets form a plug within the damaged area. Now, of course, platelets can potentially stop this just by sticking to each other if there's a very small damaged area. But generally, we're going to have a bigger damaged area, and we're going to have something a little bit more elaborate. So these platelets release signals that convert fibrinogen, one of our plasma proteins, into thread-like fibrin. So we see some fibrin here over on the left with red blood cells trapped in it. So these fibrin threads are going to trap red blood cells, and that is going to be our clot. Sticky platelets, this fibrinogen converted into fibrin, trapping red blood cells, forming a clot, sealing the hole at that area until we can actually repair the damage. Now, when we talk about clots, there are three terms that really come to mind. Those would be thrombus. A thrombus is a stationary clot. And as we can see over here to the right, what we're talking about in this case is a clot that is forming within veins. You can see these are deep veins of the leg, and we're referring to what we call deep vein thrombosis, where we are going to, near one of our valves here, form a clot, and this clot is going to build up behind this valve. Now, this can happen in superficial vessels as well, but it's kind of a major concern here when they form in deep veins like this. And the reason why is that a thrombus can become what we call an embolus. And we see an embolus here over to the right, which is a piece of clot that is broken free and is now traveling within the bloodstream. Now, as you know, as we return up towards the heart, we're going to have a progressive series of larger vessels as our smaller vessels join together. And so this embolus should be free to continue to float back towards the heart within these progressively larger vessels. But once we get to the heart and we are pumped away from the heart, we're going to go from a very large vessel progressively into smaller vessels as we approach sites of exchange. So in particular here, if we have a embolus traveling up our leg and working its way back in towards our body, it's going to travel through the femoral vein to the external iliac vein, to the common iliac vein, to the inferior vena cava. And this is going to return to the right side of the heart, so into the right atrium. That will travel down to the right ventricle, and that pumps in what we call our pulmonary circuit, delivering blood to the lungs. So as this clot works its way through the pulmonary trunk and pulmonary arteries, and ultimately progressively smaller branches within the pulmonary circuit in the lungs, it's eventually going to get stuck. And that's what we call a thromboembolism. That is, we've had an embolism, which is a chunk coming off of a thrombus. It's gone through the bloodstream until it's reached a small vessel, at which point it causes a clot, blocking blood flow and ultimately killing that area and perhaps the person. All right, our disorder associated with blood clotting is hemophilia. And this is going to be another case where we have a disorder which is actually a bunch of different disorders. In other words, a number of different ways in which we can have the same outcome. And hemophilia we recognize as being this inherited clotting disorder. As a result of having 12 clotting factors, which take part in making those steps in the clotting process, we have a lot of different genes that can have defects and result in the same inability to clot. So in the most common case of inherited hemophilia, the gene for clotting factor 8 is going to be the problem. Now, since it's on the X chromosome, and since females have 2 X chromosomes, they are more likely to have a functional copy of this gene, whereas males who have a single X chromosome, and their Y would be paired with it, are going to be more likely to have a single non-functional copy. That is because they only have one copy, it either works or it doesn't, and they are more likely to have this common inherited form of hemophilia. So let's take a look at what goes on here. Normally, if we have injury to a blood vessel, the blood vessel will constrict. We call this vasoconstriction, and we're going to enter our clotting process. So normally, we're going to have platelets form a plug. We're going to have clotting factors which are going to take part in this, and our fibrin clot forms as we get red blood cells trapped within that fibrin meshwork. But if we have hemophilia, what's going to happen is that we're going to lack a particular clotting factor, in this case clotting factor 8, and we're going to have only a weak platelet plug forming. We are basically going to, in this case, not stop the bleeding here with the platelet plug, and we're going to slowly or incompletely form the fibrin component of the clot, and that is we're going to continue to leak blood from this damaged vessel at a point in time where a normal person would have healed this. One of the common things that's going to be associated with this, of course, is an increase in bruising, the bleeding around sites of damage. And obviously damage can occur in a lot of places where people wouldn't normally notice it, such as around the joints. Okay, now we're going to get into blood typing. And of course, there are a couple of different things we want to recognize in here. We want to talk about the ABO blood typing and the RH factor. And we want to begin with this idea that blood cells have antigens on their surface. So these are going to be glycoproteins on the outside of our red blood cells, and they are going to be specific to each blood type. But the plasma is also going to carry antibodies. So here we see that we have a type A blood cell, and we have an anti-B antibody. And this is the kind of antibody that will be carried by a person who is type A. So in this case, we don't have any what we call agglutination. But if we look at the picture down below here, we're going to recognize what this agglutination is, the sticking together of blood cells. We have, again, a type A blood cell, and we have an anti-A antibody. You can see here they're listing this as being in a type B recipient who's been given type A blood. Of course, that's something we should not do, and this explains why. The type B person has anti-A antibodies, which bind to the antigens on type A red blood cells, causing them to stick together. As you can imagine, this sticking together, this agglutination, is essentially a clot formation process. And so if we were to give type A blood to a type B recipient who has these anti-A antibodies, we're going to get clotting within their vessels, and blood will not flow through capillaries as normal. Alright, so let's talk about some of these blood types then. What do these things actually have? Well, if you are type A, you have type A antigens and anti-B antibodies. So as a type A person, you don't have any anti-A antibodies. That would cause your own blood cells to stick together. But if you were to receive type B blood, you would have agglutination occur as they stick together by virtue of your anti-B antibodies. Down below, we see a type B blood. And again, I want to point out maybe that these antigens that they're showing here are representative so we can understand what's going on. It's not as if we have round or spiky ones here, but we do have different protein types. So the type B has type B antigens and anti-A antibodies. As we saw earlier, those anti-A antibodies would stick type A blood cells together, so we can't give a type B person A-type blood cells as a transfusion. Now we do have a couple more types. We have, first of all, the type AB. And you can see here on the type AB we have both the A and the B antigens. So when we are type AB, it's not a blend of type A and type B, but rather possessing the exact characteristics of both the type A and the type B. So in other words, rather than being something in between A and B, you are simultaneously A and B. This will be important for our discussion of heredity later, where we're going to again talk about blood typing, not the particular relationships here we have between the antibodies and the antigens, but how exactly it is that we inherit our blood type from our parents. We do recognize the type AB person as the universal recipient. That is, that they can receive not only type AB blood, that is type specific, but they can receive A, B, or O blood cells. And the reason for this is that they have neither anti-A nor anti-B antibodies. And of course we know that they can't, because they have both type A and type B antigens, so either antibody would cause agglutination. If we look down below here at type O is actually going to lack antigens. So it's not type A, it's not type B, it doesn't have either one of those, but it does have antibodies for both. So we recognize the type O person as a universal donor. They can donate their blood cells to anybody. Now the downside to this, of course, is that type O person needs to receive type O blood because they have antibodies against both the A and the B, meaning that they can't receive A, they can't receive B, and they certainly can't receive type AB. Now, I do want to point out that we have to account for other factors, so people don't simply give this kind of type O blood to anybody. What you could do in an emergency is you could give type O negative blood, that'll deal with the Rh factor we'll talk about next, to somebody if you didn't have time in an emergency or the capabilities to type them, but typing someone so that you can give them type-specific blood is a relatively quick process today. All right, we are going to talk about a disorder associated with the blood as well, and this is the hemolytic disease of the newborn. And so obviously this will be affecting babies as they are developing in their mother's womb. And in this case, what happens is that we have Rh positive antigens. So we're recognizing that there's a separate factor other than the A, B, and O. In fact, there are many factors. One of them is known as the Rh factor. And you are either going to be positive for this or negative for this. And we will most likely talk about how this is inherited at the same time later in the semester as talking about how the A, B, and O blood typing is inherited. So these Rh positive antigens stimulate an immune response in an Rh negative mother. So that's the important thing that's going on here. Now, the weird thing about this is we always talk about blood cells not crossing the placenta, and in fact they don't. But towards the end of pregnancy and towards the birthing process, this can certainly occur as we have, obviously the birthing process taking place. It's going to be a bloodier process than the developmental process. So what happens here, if we take a look to the right, we have an Rh positive fetus, and these blood cells are said to leak across the placenta into the mother's bloodstream. Now, because they don't tend to do this early in the pregnancy, the effect of the hemolytic disease of the newborn is going to occur for subsequent children, not the child initially that's going to have its positive blood cells leaking across the placenta. But in this case, the Rh negative mother is going to develop antibodies to that. So we see antibodies developing within the mother. And again, since this baby is going to be born before that happens, an additional baby that's in there is going to have antibodies within the mother's blood be able to move across the placenta and attack the blood cells that are within that developing fetus. So how do we deal with this idea? Well, it's treated by something called RhoGAM. This is an immunoglobulin shot, and it's basically going to make sure that the mother, who is potentially exposed to the Rh positive antigens from her child, isn't going to develop the antibodies. In other words, we're going to suppress her immune system so that she doesn't have an immune response to this child. When does this thing occur? Well, the mother must be Rh negative to form those antibodies to the Rh positive blood. And of course, in order to have a child who then is Rh positive, the father must be Rh positive to pass on those genes to the child.